My name is Brittany Choate. I'm Director of Programs for Saliva Direct Inc. And it's my pleasure to welcome you to this session of Saliva Direct's 4th annual conference. This afternoon session, or at least for those of us on the East Coast, is going to focus on patient-centered innovation in women's health. We're very excited to have a number of individuals joining us from around the world, as well as 2 amazing panelists who are going to be sharing with us their work in this very critical field and emerging industry. Each of the speakers will have about 20 minutes to share their slides, and then we'll have a live Q&A at the end of the session. With those announcements complete, I'm very pleased to introduce our first speaker today, Dr. Sofiane Bendifallah, the American Hospital of Paris. We're very grateful that he's here today to share his information about the innovations in epigenetics for endometriosis, the saliva test value. Dr. Bendifallah, over to you.
I'm very happy to be this evening with you, and very happy to discuss about epigenetics and this breakthrough technology based on the saliva test and microRNA. When I come back here 2 years ago, we discussed about this new breakthrough technology based on the saliva, and 2 years after, I'm very proud and happy to share with you where we are and when we stand in 2025. This is my declaration of interest. The most important fact is behind this breakthrough technology of early diagnosis for endometriosis that we have patients, and the role of this new technology is to be able to overcome some challenge associated to the diagnosis. As you know, because the endometriosis is the most complex disease, we have no pathological sign associated with this disease. We have many imaging limitation, especially for the most early stage. And because of this uncertainty, we have some delay associated to this point. When we discuss about, and more precisely about why endometriosis is so complex to diagnose, especially because we have different phenotypes. Among those, we have this peritoneal endometriosis phenotype, which is based on the most complex diagnosis. And these lesions are very small, and that explains why, based on imaging, we are not totally able to diagnose it. In fact, when we have this kind of lesion, it is also difficult to be able to say we have endometriosis, and that explains why, with the delay of diagnosis, we'll have some impact of the disease, impact of the symptom on patient, and that explains why this disease is so impactful for patient and have so many consequences on daily life. We also have impact on chronic pelvic pain associated in this lesion will have an impact on this chronic pelvic pain and on the central nervous system, which may explain all the consequences. At last, we will have a disease very frequent with an impact on mental health, an impact on education, an impact on work, and a social cost. And because of the early diagnosis is so important, it was explained, especially because now we have some data which make the relation between the disease and the consequences on several risks, risk of cancer, risk of stroke, and that explains why it's so important to diagnose it. But now, in the last decade, this very painful disease was treated especially empirically by hormonal treatment. And the treatment success or failure neither confirm or exclude the endometriosis. And that's why now the issue, and the only issue, is to be able to diagnose the patient early and to be able to offer them new prevent care pathway and new prevent treatment. And in this issue of pain, we have also another issue which is called infertility. And for this patient with this lesion, with this disease, we will have this impact on infertility. And that explains why it is so important to diagnose the disease very early. But now, this delay in diagnosis was well known, was well described, and that explains why we have many research on this point. And the only way to be able to reduce this delay is to be able to add new breakthrough technology. This is why we work on this microRNA saliva test signature, to be able to add this epigenetic background to the disease and to be able to detect it very early. But now, why it is so important to identify this peritoneal form? I will show you some lesion. Like here, you have the early window where we are able to diagnose the disease. We had to understand that at the early stage, since the teenage period until 25 years old, the most important frequent lesions are peritoneal. And this lesion If you look well what happens in practice, this is different clinical situation associated with this lesion. And if you look well what happens in reality, all these described lesions are endometriosis, and all are under the capabilities of imaging to be diagnosed. And by surgery, here, if you look well, diagnosing endometriosis and peritoneal lesion is like a real challenge. I will show you some screen about surgery, and for example, this one. This is what happened in terms of surgery. This is a pelvic video of endometriosis patient, and I will show you what may be a lesion with endometriosis. This is this small lesion, and all here we can understand that this small lesion can be underdiagnosed and can be under-treated. That explains why we change the paradigm of diagnosis and we work on the biology and especially on epigenetics to be able to understand more and more precisely why and where can be the disease. If you take some time to understand what happened in the last decades. In the last decades, if you was a woman with pain with normal imaging, the majority of these patients underwent surgery, but you need to understand that even if we perform this surgery, we will have this discrepancy between the pain and between the capability of this surgery to understand well the disease. And even we perform surgery, we have sometimes some discrepancy in the diagnosis. And in surgery, not seeing a lesion does not mean there is no endometriosis. And this explains why this surgery is not and can be the gold standard for this patient. But now, what is the value of epigenetics in 2025? This value is now very clear because, as you know, for endometriosis, which is probably most epigenetics than genetics, we have this microRNA and more globally the RNA, which is these few molecules which have the capability to describe the real phenotype of the disease and to understand, thanks to this microRNA, what happens in terms of regulation and of mechanism and physiopathological mechanism. These microRNA, and especially in endometriosis, have this capability to describe all the mechanism and all the pathological mechanism of this disease. Thanks to this epigenetics background, thanks to the capability to detect this microRNA and more globally this RNA, we have now this capability to understand the regulation, the dysregulation of this mechanism, and to be able to overcome the limitation of previous clinical sign, and previous imaging. This is what this is the scientific background uh, based on the saliva, and all the data have been published since now several years, and more recently we have now promising application of saliva biomarker, especially in clinical diagnosis. And the saliva in this issue is very prodigious. Why? Because in the saliva we have this capability to detect in less than 2 milliliters of saliva all the panel of biomarker associated to a disease. And this panel have the capability to be understood thanks to new technology like AA. And if we have this capability to detect all these microRNA, we have the capability to understand well the disease. This background is well known in cancer, is well known especially in gynecology for ovarian cancer, and this is why we work 4 years ago on this issue and how can we add this technology to endometriosis care pathway. If we come back now to the most recent work associated to this saliva microRNA testing, we have now more and more data exploring the capability of saliva and microRNA to detect in many lesions, many conditions, cancer, Parkinson, infection, traumatism, the capability to understand well the disease and the phenotype. If you remember where we stand now comparatively in 2022. In 2022, we were, for example, this publication from Monica said that for the moment, no paper examined the role of microRNA in the saliva. and there is no scientifically proven biomarker for endometriosis. This is what we had in 2021. Since this moment, we specifically work on this saliva test. We perform one development study, one validation study called 1000 study, and all these data have been now published. And I can say that I'm very proud to say that 4 years after this moment and this publication, we use the saliva test in practice and in clinical practice in 100 centers in Paris. I can also say that I am very lucky to be able now to use it in my clinical practice, and how it's happened now in clinical practice in France. In France, now, if you have pain, if you are a woman with pain, with normal imaging, you have this capability to obtain this saliva test and to do this test. And in my practice, I observe several value for this test. First, now is accessible to all my patients. Second, with this capability to detect early the disease, thanks to this biological paradigm, I am the capability to reduce the delay and to be able to overcome the undiagnosed and untreated women. I have also the capability to start very early all the care pathway dedicated to reduce the pain and to reduce the impact of the disease on the patient lives. I have also now with this early detection have the ability to prevent infertility associated to endometriosis, and this is what I observe in the last years using this test in my practice. But now, in terms of biology, in terms of technology, how it works. The test is based on microRNA, is based on signature and AA, and this test is now have this ability. This is how it works in practice. I will show you this video, which show you how we receive the saliva in this small sample, and how we have the capability to, in 2 milliliters of saliva, to obtain some liquids, and in these liquids we have this capability to detect all the panel of biomarker of all RNA, and in this RNA the detection is based on microRNA because the saliva is based on this microRNA signature. The full automatized care in this full automatized background have the capability to transform few milliliters of saliva in fields of 4 giga of data, and these data, thanks to the artificial intelligence, have been analyzed and have been estimated as associated with endo and not. And this is exactly how we use it now. And I would like to share how we plan to use it in clinical practice. Now, if you have endo, if you have normal exam and pain, if we are not able to detect endometriosis, we perform the saliva test. If you have sign of infertility, we have to perform this endo test, and this is exactly the new algorithm of detection for endometriosis in France. To conclude, I would like to show you that this on the test have this ability to reduce the delay and have this ability to early diagnose the patient and have this ability to design a new care pathway for all these women. And this is for me 4 years after the first publication of the saliva and the value of the saliva in endometriosis, real success for us. And behind this success of technology, of biology, of epigenetics, we have patients, and we have this capability to now give a real and accurate answer for all the patients with pain. Thank you very much for your attention.
Thank you, Dr. Bendifallah. That was fantastic to hear the update and how much progress there's been made since the last time we were able to hear from you. With that, I'm very excited to introduce our next speaker for today's Women's Health Centered session. And this is Søren Therkelsen joining us from Qvin. And he's going to be sharing with us about the future of diagnostics, unlocking the power of menstrual blood.
Thank you so much, Brittany. And it's really exciting to see Dr. Sofiane's work. As someone working in the industry, I cannot express the excitement on behalf of women's health and on behalf of women. I know how difficult it is to develop tests, and endometriosis is an extremely complex disease. To be able to do this from saliva is mind-blowing, and what you've accomplished is just outstanding. So I just want to congratulate you. And thank you to Saliva Direct for this opportunity to speak today. My name is Søren. I'm a co-founder of a company called Qvin. Qvin means woman in Danish. I'm from Denmark, and at Qvin, I'm the co-founder and chief product officer. And I'm going to talk about menstrual blood and what menstrual blood is and how we can use it. But first, a disclaimer. I'm obviously a man. I don't menstruate. I don't have that experience, but I've spent a lot of time with women and been trying to understand the physiology and everything that goes with it. I'm a product professional. I come from a product mind. That's what I do for a living, and so I develop scientific tools. And then I'm the co-founder of Qvin, which means I have a stake in the company. And my co-founder Sarah is an MD pursuing a PhD in menstrual blood. So we have someone at the helm that's very scientifically advanced to support my product work. And I'm here to explain the product experience and the science around menstruation objectively, but obviously as someone that's building my own thing, obviously I'll have a little bit of preference. So Qvin is building around menstrual blood, and it has a wide range of opportunities to impact how we diagnose conditions. In fact, billions of women globally are affected by a number of conditions that lack good diagnostics, like endometriosis. And this is what we as a team set out to try and solve and work with using menstrual blood. And so menstrual cycles is a regularly occurring event for women. And what we focus on is the beginning of the cycle, which is the release of last cycle's endometrial lining. It's coming out with blood, and through the vagina, you pick up a lot of different things. And so this is the sample type we're interested in. And it has a lot of opportunity. You have 2 billion women who menstruate monthly. In fact, 300 million women are probably menstruating this very second. It's kind of staggering how much it is and how little it's talked about. And if you utilize the sample type, you have an opportunity to screen and monitor conditions for a total of 35 years on average. And for women, they use a lot of different products during those years, up to 11,000 pads and tampons usually. And menstrual blood offers this non-invasive sample opportunity, just like saliva does. But menstrual blood composition is different in the sense of you get 3 different sample types in one. You get peripheral blood without using needles. You get access to cervicovaginal mucus and secretions as it travels through the vaginal canal. And then where it originates from, you get some endometrial cells and fragments, which normally would be collected with an endometrial biopsy. So you really have a non-invasive sample type that is very, very rich in information. And when you consider that opportunity, it is largely an unexplored opportunity. If you go and I just did some PubMed searching just to kind of get a grasp prior to this, like how much do we actually study this? There's less than 100 publications on the utility of menstrual blood from a diagnostic purpose. But if you start looking into that, well, then you start seeing it has a lot of promise within a number of very important health categories, including endometriosis. And we've done a lot of work in that, and I will share some of our insights. But why is this research gap here? What has happened? And there's a multitude of reasons why we haven't studied menstrual blood. First of all, women historically has been underrepresented in studies. In fact, there were a lot of time they were excluded from studies altogether, simply because female biology was too complex to include into drug development, for example. We didn't like those pesky hormones. There's been a giant lack of funding just in general. It's definitely improving as of late, but that's one of the reasons. There's really a lack of regulatory clarity. How do you even leverage this sample source? We as a team had to fight that with the FDA and really work with them to understand how do you bring a test to market. And then when you study menstrual blood, the study designs can often be impractical simply because of the nature of what actually happens during menstruation. I'll come back to some of that later in the presentation. And then we, as an industry, just lack tools and protocols. Like this hasn't existed. And so we've spent a lot of time covering all of these as a team. And I'll show you how we build tools. So I'm going to focus on collection today and tell you how collection has to be done and how you what you can do with the samples once you get them to the lab. So first of all, to collect menstrual blood, you have to appreciate that it's a really dynamic collection in the sense that it happens usually once per month for a period of 5 days. And it's not like you walk into a place and then, like you do with a saliva, you just sit down and spit in a tube and it's done, or you come in for your blood draw and it's done. No, here it's something that happens over a duration of time, and it's usually happening while the woman is doing a lot of other activities, sleeping, working, taking care of offspring, or doing activities. So when you collect, you sort of need to fit into this life of a woman. And when she's on her period, the sample volume fluctuates quite a lot. It usually picks up in the first couple of days with a strong flow, and then she consumes more products when she does. And it's often a mix of products that she compiled depending on how strong her flow is. So to talk a little bit about products used globally, the category most women use is pads, menstrual pads. It's by far the most used type of collection in the world, really to prevent spillover, or how to explain that, the mess that's associated with it. Pads in low and middle income countries, it's by a far magnitude the primary product used. Tampons is a more or less a Western phenomenon. We use them in North America or in Europe, where they're also used a lot. And cups, being a relatively new phenomenon, its usage is increasing, especially among younger and eco-conscious users. But that actually, as a collection device, offers a lot of utility. And when you consider the different ways you can collect, they have some advantages and disadvantages, each of them. Starting with pads, they can be used by anyone. It's not a limitation. If you're used to using pads or tampons, tampons or cups, you can still use a pad. So anyone can really do it. It sits externally to the body, which means that the fact of inserting something vaginally for some cultures can be a challenge. And then some of the disadvantages, you can really collect only during periods, and some users prefer other methods, and people sometimes swear to their specific method. Tampons are great in the sense that you can use them anytime, which means you can also do it in between periods or after menopause. Might not be the best user experience, but you can still collect samples from the vaginal tract that way. It wouldn't be blood unless you have abnormal bleeding, but it definitely is an advantage. And they're discreet, and some women definitely prefer that. The flip side, again, some users are not comfortable, and often it requires a buffer to stabilize the sample. Once it comes into a tampon, it soaks in, usually to a cotton material. In order to be good for laboratory testing, you have to preserve it in some preservative, and that can limit actually your different analytical opportunities, because you end up designing the buffer to that test, and so it's not as universal of a way. And then actually also shipping buffers through mail can have some flammable. They're often flammable, and there's risks associated with that. Cups are great. You can collect liquid samples. You can collect cells, which is really interesting and has a lot of science opportunity. But they have low adoption, and the sample stability is super short if you use them for laboratory testing. So because sample stability is short, you would need the sample back, usually kept cold. So you need some ice packs, which is logistically challenging for a person to have at home. You need to ship them overnight, which is logistically difficult and very expensive. And so that's really a drawback that makes it much more expensive to work with. There are multiple entities working on menstrual blood collection for laboratory testing. Ourselves, a team called Torelli, that are working on an ELISA test, more of a strip-based system, so it's more of a point of care. You can do the test at home that way. Next-Gen Jane and they are working on tampons. Next-Gen Jane is doing amazing work in endometriosis, and they a lot of good work in HPV and STIs. Tulipon is really interesting. They have this sort of a hybrid device between a tampon and a cup. I think they're doing great. It's a really interesting device they've created. And the Blood and cup company is expanding what you can do with the liquid sample. And so I'm, from Qvin, and we've developed the QPAD. So I wanted to share more about some of the product requirements that really informed how we built our product. And so first of all, we put the patient in the center. And when you work with the patient, it has to be super, super easy to collect. And the instructions should be simple. That's really key. And you have to fit the product into the schedule of the woman. So it has to be an at-home collection. You have to accept that, and it's nothing. You can't just go in anywhere and get it done. So it's just very important that you design your product accordingly. And for the laboratory to be used, you really have to figure out sample stability. So for us, we used the goal of working with ambient shipping temperatures, which I also think you do a lot with saliva, and I think that's really the way to go. It keeps it much more affordable to work with your samples. And the laboratory testing should be easy, and should ideally support both qualitative and quantitative testing. And you have to make it cheap. If you make your product too expensive, you can't get the global adoption that you might be looking for. For us, our vision is to make products available for every person on the planet or every woman on the planet. And that's not going to work if it's hundreds of dollars. So you really have to use inexpensive materials. And then through painful learnings, data is at the center of everything. And so you really have to have end-to-end traceability around all the data that's associated with your devices, your patients, the laboratory, trials, biobanking. All of this needs to connect end-to-end, and is a key. Yeah. And so I wanted to show the QPAD, what it looks like, how it works. It's basically a pad. It's has this green oval printed on the top, which is an indication for when she's done collecting her samples. She pulls out this strip, which is a dried blood spot strip, and places it in this container, which is loaded with desiccant and keeps the sample dry during transportation. The envelope is pre-stamped with shipping information, and as the phone is really central to the experience, she registers the kit on her phone, and she also can receive the results on her phone. So that's also part of the user experience. This is how it works from a patient perspective. The device itself, as I mentioned, is a pad, organic cotton. It had to pass various biocompatibility certifications. The strip was designed with Whatman paper. So if you guys have worked with neonatal screening, it's the same type of paper that's being used to collect the sample. And every device has unique ID labeled on them from the point of manufacturing. So we keep that back to that traceability we discussed earlier. And then the sample container was a hard requirement too. We have a lot of desiccant inside where there's room for 2 strips. Basically, a woman collects 2 samples in one go. And that container also protects it during transportation, so it won't break. It protects it against humidity, and it can travel during ambient temperature really safely. And when the sample arrives in the laboratory, they come back on these strips. And if you've again worked with neonatals, you will know the procedure. But you basically punch out of this the strip. I show here the front and the back of what a sample may look like. And these little pallets here you put into a tube, you reconstitute the sample. This is very simplified what I'm explaining, but you may have a very specific protocol per test. And you can use your whichever analyzer you're really looking for. Well, analyzers, I'll come back to analyzers in a minute. And then I mentioned this traceability aspect. And so we've built this really advanced software system which have everything from raw materials, when you put together everything that goes into the device, all the way to the journey until it makes it to the laboratory and you biobank it. Every state, every interaction, everyone who touched the sample, everything is linked and kept track of in this really neat system, which is very powerful for the way when we run our science. Because we work with dried blood spots, you really have access to a large range of different analytical methods. And sample stability on a dried blood spot is long compared to working with liquid samples, for example, liquid blood. I wouldn't know for all the different methods for how long it is for saliva, but I imagine it's somewhat similar. Even though, of course, every stability is measured per test, but in general, the numbers on your right is to give you an idea about how the estimated stability time if it's kept desiccated and at room temperature. This is not a one size fits all. There's a lot more to it, so that's just a general information. So we spent time trying to develop a proof of concept test to take it all the way to the market, and we developed an HbA1c test used for monitoring diabetes. And what you see on the left is Qpad collected menstrual blood on the left axis, and on the bottom axis you see venous blood, and you see really, really tight correlation, an R squared of 99.5. So there's a really, really strong correlation between the two sample types. And when we received our FDA clearance, we developed a new, they gave us a new product code called QZG. It's the first product code in the world for menstrual blood collection devices. And when we received the clearance, we also received an over-the-counter clearance, which means this can be acquired without prescription. So you could, in theory, buy this in your local pharmacy or on Amazon or wherever you want. When we developed this test and had to take it through the FDA, there's also a whole range of different analytical studies you have to conduct, as you guys know as laboratorians. And some of those I will highlight here. I would say really key one, it was like the shipping stability, really showing that the sample would remain stable under various conditions. And so we obtained a 28-day stability claim. Interference was super, super hard. And I think it's hard also for everyone working in saliva. I think the advantage for saliva is that interference has at this point probably been more established from a regulatory perspective, even though I understand that there are so many different things that a person can put in their mouth and which may interfere. Negotiating that with the regulatory body on what that may be, the concentration, how often, and how long can they have had it in their mouth. There's so many potential risks and materials that can come into play. Similarly, it's probably worse actually for saliva, but in menstrual blood, we had to show various common endogenous and exogenous sample types of substances, and so sweat, urine, feces were part of our semen, were part of our testing. We had to show deodorants, wet wipes. And so developing that list and confirm it with regulatory was very, very difficult. And then develop the protocol afterwards to show it in the sample was a really, really hard challenge for us. But we figured it out, and now we sort of have the protocol. We can do this again, and so that's the great news. And then usability is also something I'll highlight. We're very proud of that 97.5% of our users collected samples successful in the first attempt, which I find quite good actually. I don't know what it is for saliva. It's probably similar, but I'm very proud of this. It has been a lot of work around the actual product experience and the instructions to get to this point. We've done a lot of work in comparing menstrual blood to blood or serum, and so here is a subset of different tests. So you can see there's a relationship between menstrual blood. This is one of our first publications between menstrual blood and venous blood. I actually think this was done using a menstrual cup when we did it. We then continued with Qpad and dried menstrual samples, and we started seeing sample stability for a minimum of 2 weeks across biomarkers. And we also learned that the day of period really didn't matter for the performance of these specific tests. It may be different depending on what you're looking for, of course. We've also done work to understand the cytokine profile of menstrual blood versus normal blood, and we saw a very different cytokine profile with elevated cytokines for a number of cytokines linked with endometriosis and endometritis, so that's just some of the insights that we can do with this analytical method. we are interested in accelerating our research, and we're interested in working with anyone who finds this interesting or want to work on menstrual blood. We make our devices available for every laboratory that are interested. You can reach out to me here, or you can scan this QR code, and someone from our team will reach out to you to discuss your objectives or your studies. But with that, I thank you for your time today, and I look forward to a discussion.
Thank you, Søren. Very much appreciate you sharing about the technology your team is innovating and also that invitation for collaboration. That's something that we really appreciate about this conference, bringing together different perspectives, whether it's saliva or menstrual blood or other non-traditional sample types, and creating a space where we can exchange information, share, and help each other get across those finish lines. For anyone who hasn't had the opportunity yet but would like to, please remember to use the Q&A function down in the bottom right-hand corner of your screen to submit a question to our panelists. While we're waiting for additional questions to come in, we've got some that are available. The first question that came through is for both of you, so Søren and Sofiane. Would you each mind speaking to the regulatory hurdles that you are facing or have faced to bring these tests to market? So Søren, since you're already on camera and on off mute, would you mind taking that question first?
Yeah, I mean, it was super difficult, and it's also an environment that we're a U.S. company, and it's a regulatory environment that's changing. The FDA was hampering down on CLIA, and it really impacted what you could do with LDTs. And we actually, in our own CLIA lab in California, had developed 6 different tests, which were prohibited to launch after we got clearance. Has been a huge setback for us. We basically view this as we have to take every test through the FDA, which is very expensive and complicated to do. But we are up for the challenge, and we focus now on tests that are high impact, high reimbursement, focusing on, among other, HPV and STIs, but also these conditions like endometriosis, which really can impact people and have better pay. And so, but on regulatory, it was so hard because the FDA had never seen anything like this, and so it was like educating them really, and that was hard because they was like, you know, I'm a man, so I should not discriminate against my own gender, but there was a bunch of men that didn't understand what the hell we were talking about, and there was just like a lot of the time irrelevant questions. You're like, that's not how the physiology works, you know. It felt like you had to be so, yeah. That was definitely hard. Yeah. I don't know if that gives some color.
Oh, try that again. Would you mind sharing, Sofiane, about the regulatory hurdles that
Brittany, you are on mute. Yeah.
Are currently facing?
It's always, you know, it's complex to bring to the market some breakthrough technology, because you have lots of limitations associated to the mindset of the people, to the mindset of the regulator, to the mindset of also because this is science in terms of science and in terms of reproducibility, and specifically for the blood in a tampon or the blood in cups or the saliva in sample, we always need to bring many explanation and many example and data before to change this mindset. And for me, this is the major complication to bring this and to change the mind of the regulator.
Absolutely. Introducing those non-traditional sample types, you have to prove your case and prove your science. You have to really be a pioneer in those aspects, and appreciate the work that both of you are doing with your teams to do that. Another question that came through is regarding the patient experience. How did each of you, either in the past or are currently seeking input from patients to confirm acceptance and usability from a patient perspective?
For the saliva, it's very simple. I can't say nothing more, because you just need to split on 2 milliliter of saliva. You have to, of course, you have to respect some condition. You have to respect some facts, but it's very easy in practice. And we have less than 1% of all the sample. We are not useful, or we can't detect any microRNA, but usually it is because the people have take food before, or have drink something before, and this is the condition. If we remove all this, the saliva is very simple.
Beautiful. Yeah, that high usability rate is something that, Søren, you featured in your presentation as well. Would you mind speaking to how you were doing those confirmation studies with individuals using your screening protocol?
Yeah, I mean, there's I think the blessing of Saliva is that it's been known for a long time, and the collection devices have been developed by companies independent, so you can focus on your science and develop a test. And for us, we had to start with a collection device and to build it from scratch. And so we started with a cup, it's just very practical, and just to collect the sample and see what the sample was, because there was no science. So we collected, and we learned from using cups that was just impractical. Like the samples weren't stable, users wouldn't use them. So we realized that and accepted that we had to switch to a pad. Was like the widest use. And then we came across dried blood spot and thought that made sense, but we had to build a dried blood spot into the product. And so I stood at my kitchen with scissors and paper and taped it on and gave it to my friends, and learned from that what's going on. Oh, it's pointy, or it's sticky, or you don't know when it's enough. And iteration after iteration, and we had to find a pad manufacturer, because that we needed to build this into a pad, and so that was hard. And working with them, and found a really great team in Italy, and collecting enough sample. And when is enough? How do you know? I mean, if you spit in the tube, you have enough. You can just look at the line. But here, it's something that happens over many days or many hours, maybe. Sometimes it can go down to 10 minutes if you're a really heavy bleeder. So really being close to your patient, really having a lot of communication, really listening back and adjusting your experience. It's a mix between, your product, actually what it looks like and feels like, and that input. And you have to have close loops between the two and iterate fast. It's challenging because people only menstruate every 28 days. So, you can't just, hey, try this new one. You got to wait, and then they forget. And, so I would say it's really difficult. But yeah, you just have to listen to them, and then you will eventually get there. And then eventually we had usability studies and passed them, and I'm very proud of that.
That's fantastic. I think with both of you having such high levels of patient compliance and having viable samples, it shows the testament of just how effective Saliva is as a sample type, and also all the work that Søren and your team has done to ensure that you've got appropriate volumes and collections and things like that. Those elements of making sure the patient experience, that it's as user-friendly as possible, is really critical to a lot of the work that our team does, and also others who have been joining this conference as well. Another question that came through is, in both of your presentations, you were highlighting a number of different targets or potential future applications. And so there was some curiosity about, of those things you're exploring or considering for the future, what is it that you're most excited about and think could be most disruptive to the industry that you're each in? Søren, would you mind going first?
Yes. So we spend a lot of time already in blood and trying to compare ourselves to blood. And I think we are scaling back our focus on dissecting the same things in blood, simply because there's a lot of opportunity already, or a lot of companies already having tests. So trying to develop in that area doesn't make as much sense for us. The uniqueness in menstrual blood is probably mostly actually all the way up from your uterus, or the uterine lining. It's really hard to get access to samples here. And I think I'm really excited about the opportunities to understand infertility better, and really dive into maybe the reasons of endometriosis, adenomyosis, fibroids, cancer, uterus cancer, uterine lining cancer, ovarian cancer. We're so close to proximity of issues, and the sample is so readily available. And the good news for us is that we've developed the collection device, the container to acquire the sample, and it's really more about now discovering what can this actually do. And that makes me smile every day, and it's like what I've been waking up for the last 11 years, just thinking about what can we do for women. And I'm really excited about the potential for specifically infertility and cancer. I think that's really exciting.
I can hear and see the excitement that you have and the passion that you have for this kind of work with how you're sharing that information. Thank you. Sofiane, what about you and your team? What kind of applications are you most excited about?
What says Søren now, it's totally the same for us. We also have the same excitation, because he said we have the device now, and we have this ability to detect many million of biomarker. And for the saliva, it's more specific and more associated to the RNA and all the panel of RNA. And as you know, in RNA, we have microRNA, RNA messenger, RNA circular, RNA transfer, and so on. and we have this ability now to work on what is the evolution of the disease, how the patient may respond to the treatment, how can we define the real phenotype and biological phenotype of the disease in endo. This is exactly where we are, and this is exactly where Søren is also, and it's very exciting. It's very exciting.
Absolutely, absolutely. Who knows? There might be some sort of a partnership or opportunity for comparisons. Maybe not sharing sample types, but opportunities for working together. Or maybe you do start sharing sample types. Who knows? That leads into, I think we've got time for one more question, and that is kind of thinking about these future applications and these partnerships and things like that. What do you each think is the timeline for the technology you're working on to become more common in the marketplace, to become part of those annual well women checkups or get used in screening? Do you have optimism for them being readily integrated? Sofiane, would you take this one first?
I am always very optimistic, and it depends on one fact: if the women are able to change and the world. It's very easy. We have now technology, we have the background and the science behind all this technology, and if these tools are able to change the daily life of this patient, I am very optimistic. Less than 5 years.
Less than 5 years. Amazing, amazing. Søren, what about you? What's your perspective?
But I'm more optimistic for Sofiane than he is for himself, because he's already in the market in France and doing it. And so I just congratulations. I mean, it's very hard to do scale, and distribution is a challenge. The traditional way of getting access to testing is through your physician, or your provider. So whenever you develop a test, and that's great and it looks good, well, you have another hurdle, which is insurance and pay. What's the reimbursement, and then you have education of physician and adoption that way. And there's a lot of education that needs to happen. So that's ahead of any test when you develop it, especially with a new sample type. I think for us, the massive opportunity for mass adoption is what we're focusing right now on HPV and STIs, because those are prescribed globally, and we all have a need to detect cervical cancer and infectious diseases. They're reimbursed really well, they're understood, and collection devices such as vaginal swabs are making it into the market, they're getting approval. And so we hope to ride that wave, and are working directly with manufacturers. It's a sort of a new development for us, is that we used to do all the work in-house and designed everything. And the reality is that when you work and you make it to market, you also have to distribute. And so for us, we're realizing we need to be closer to the analytical instrument manufacturers and partner with them directly on their assays, and basically putting menstrual blood as a new sample type on existing tests is a way to mass distribute and use existing laboratory infrastructure and distribution with those partners. That's for the low-hanging fruit when you already have a test. For like developing an endometriosis or a cancer test or uterine fibroids, it's more complex because you probably initially have to go through a specialist. Like Sofiane and his team are talking directly to people working in infertility or endometriosis. Like, that's how it starts. But I envision for Sofiane and team as well is that one day this would be a preventative opportunity for people to ask for if they have unexplained pain and they don't have to walk around for 10 years with this pain. They should be able to get this test, do it at home, and not ask for permission. That's the future we need to live in. And that's the advantage of saliva, menstrual blood, urine, stool, and so forth, that we don't need a professional to collect a sample. We can actually use the patient.
Thank you. Appreciate that perspective and the level of enthusiasm and inspiration that both of you bring to this, and where we're going in the future, and how each of your teams are respectively helping us get there and advancing women's health. Want to take some time again to thank both of our speakers, Søren Therkelsen and also Dr. Sofiane Bendifallah, for joining us today.